What if I told you that scientists just created life forms using artificial intelligence? No, this isn't science fiction. It happened right now in 2025, and what you're about to discover could change everything you think about biology. Picture an ordinary night in a California laboratory. Researchers stare at Petri dishes waiting for results. Suddenly, they witness something extraordinary: dead bacteria. But not for any common reason. These bacteria were eliminated by viruses that never existed in nature. Viruses whose genetic code was written by a machine. This isn't theory. This actually happened. And today, you'll understand how we got to this point, what it means for the future of medicine, and why some of the world's greatest scientists are simultaneously fascinated and concerned. But before we dive into this revolutionary discovery, you need to understand one thing: the boundary between what's natural and what's artificial just disappeared. And this has consequences you can't imagine. Let's get straight to the point. A team of scientists from Stanford University and the Arc Institute, both located in Palo Alto, achieved something unprecedented. They utilized artificial intelligence to create completely new genetic codes for viruses. And not just create on paper, these viruses actually worked. The researchers chose to work with a specific type of virus called bacteriophage, microscopic organisms that attack bacteria. The chosen target was FEX174, a relatively simple virus with only 11 genes and approximately 5,000 DNA letters. Here's where it gets interesting. They used an artificial intelligence called EVO. This AI operates on similar principles to ChatGPT, but instead of being trained on texts and articles, it was fed genetic information from about 2 million different bacteriophages. After the AI proposed 302 different viral genome designs, the scientists did something bold. They chemically synthesized these DNA sequences and introduced them into E. coli bacteria. And then came the moment of truth. What happened that night in the laboratory left even the most experienced researchers speechless. But to understand why this is so revolutionary, you need to know what makes these viruses so special. Stay with me. Out of 302 attempts, 16 worked perfectly. These computer-generated viruses began replicating, invaded the bacteria, and destroyed them completely. Brian High, the laboratory leader at the Arc Institute where the research was conducted, described the moment. It was absolutely striking, simply observing this AI-generated structure. They even managed to photograph these tiny viral particles under a microscope, small fuzzy dots representing something never before seen in Earth's life history. Jeff Boeke, a renowned biologist at NYU Langone Health, called this an impressive first step toward AI-designed life forms, and he was surprised by the machine's creativity. The AI didn't just copy existing viruses. It created variations with novel genes, truncated genes, and even rearranged gene order in ways scientists didn't expect. But wait, is this really AI-created life? Technically, no, and here's why. Viruses aren't considered living organisms in the traditional sense. They're more like rebellious fragments of genetic code with relatively small genomes and simple structures. They don't eat, don't breathe, don't reproduce on their own. They need to invade living cells to multiply. They're like biological computer programs, powerful, but not exactly alive. But if this is just the beginning, what comes next? The answer involves billions of dollars, a Nobel Prize, and a global race that could determine medicine's future. Keep watching. This didn't happen in a vacuum. We're living through a historic moment where artificial intelligence is completely transforming biology. In 2024, scientists won the Nobel Prize for using AI to predict protein structures. Investors are pouring billions of dollars into companies using AI to discover new medications. The same week this research was released, a Boston company called Lila raised $235 million to build fully automated laboratories operated by artificial intelligence. J. Craig Venter, a pioneer who created some of the first organisms with lab-produced DNA nearly 2 decades ago, offers an interesting perspective. He says AI methods are just a faster version of trial and error experiments. When his team managed to create a bacterium with a synthetic genome in 2008, it took years of manual work, testing different gene combinations. We did the manual version of AI, combing through scientific literature, leveraging what was already known, Venter explains. But it's exactly this speed that changes everything. What used to take years can now be done in weeks or days. AI can test millions of combinations virtually before scientists spend a single penny on real experiments. And the speed has practical applications that can save lives today, but it also creates risks that keep scientists awake at night. I'll explain both. Let's talk about real applications. Viruses that attack bacteria, bacteriophages, can be used as medical weapons. Doctors already attempt something called phage therapy to treat patients with severe bacterial infections that resist antibiotics. Imagine someone with an infection that doesn't respond to any conventional medication. Personalized phages could be the last hope. And it's not just human medicine. Farmers are testing phages to combat plant diseases like black rot in cabbage caused by bacteria. Samuel King, the student who led the project in Hai's laboratory, points out another crucial use. There's definitely enormous potential for this technology. Most modern gene therapy uses viruses to transport corrective genes into patients' cells. If AI can create more efficient and safer viruses to do this job, it could revolutionize treatments for genetic diseases. Biotech companies already have their eyes on this technology. Jason Kelly, CEO of Ginkgo Bioworks, a cell engineering company in Boston, believes automated laboratories where genomes are proposed by AI, tested, and continuously refined could become reality soon. This would be a national-scale scientific milestone, since cells are the building blocks of all life, Kelly states. But here's the problem. The same technology that can save millions of lives can also create threats that never existed before, and some scientists are sounding the alarm. The Stanford researchers were careful. They deliberately didn't train their AI with information about viruses that infect humans. But this precaution doesn't eliminate the risk. Other scientists, out of curiosity, good intentions, or even malice, could apply the same methods to human pathogens, and this opens completely new dimensions of danger. J. Craig Venter is direct about his concerns. One area where I recommend extreme caution is any viral enhancement research, especially when it's random and you don't know what you'll get. And then he goes deeper. If someone did this with smallpox or anthrax, I would have serious concerns. Think about it. Smallpox killed hundreds of millions of people throughout history. It was eradicated in 1980. But what if someone used AI to create a more lethal version, or more resistant to vaccines? This is the classic dual-use technology dilemma. The same tool that can cure diseases can be weaponized. And unlike nuclear weapons, which require massive resources and complex infrastructure, biotechnology is becoming increasingly accessible. Small laboratories, even talented amateurs, could have access to these tools in a few years. This raises profound questions. Should we regulate this research? How? Who decides what's allowed? And how do we balance the potential to save lives with the risk of creating new threats? But the story doesn't end here. The next step is even more ambitious and controversial. Stay until the end. The big question now is, can AI create genomes for larger, more complex organisms? An E. coli bacterium has about 1,000 times more DNA code than the FIX174 virus used in this experiment. Jeff Boca puts it in perspective. the complexity would skyrocket from impressive to far, far more than the number of subatomic particles in the universe. And there's another obstacle. While some viruses can boot up just from a synthetic DNA strand, this doesn't work for more complex organisms. To create a bacterium, a mammoth, or an AI-designed human cell, scientists would have to gradually modify an existing cell through genetic engineering, a process still extremely laborious and time-consuming. But this isn't stopping attempts. Jason Kelly, from Ginkgo Bioworks, believes automated laboratories can dramatically accelerate this process. He even frames it in geopolitical terms. The United States must ensure we get there first. This suggests there's a global race happening behind the scenes. China, Europe, United States all want to lead this biotechnological revolution. If we can create completely AI-designed cells, the implications are unimaginable. Human organs grown on demand for transplants, bacteria designed to clean pollution or produce clean fuels, agricultural crops resistant to diseases and climate change, personalized treatments for cancer and genetic diseases, but also potential undetectable biological weapons, organisms that escape control and disrupt ecosystems, ethical questions about modifying human life. So where does this leave us? We're at a historic moment. For the first time, machines can create functional genetic codes. This is no longer science fiction, it's reality. The AI-created viruses that killed bacteria in that Stanford laboratory are just the beginning, a first step on a journey that could take us to extraordinary or dangerous places. Technology isn't good or bad by itself. Everything depends on how we use it, and we as a society need to decide: how do we regulate this research without stifling innovation? How do we ensure benefits are shared globally? How do we prevent malicious use without creating insurmountable barriers to progress? The boundary between natural and artificial has disappeared. AI isn't just analyzing life, it's beginning to create it. And this raises the deepest question of all. When machines can create life, what does that mean for us, humans? The answer is still being written, and you just witnessed the first chapter. If this video made you think, leave a like and subscribe to the channel. In the comments, tell me, do you think we should continue developing this technology, or are the risks too great? I want to know your opinion. And if you want to understand more about how AI is transforming science, watch this video here. See you next time.